Hello, everyone. Welcome to week number 5 in Science 200. We are past our halfway point now in the term, so everything is downhill from here. So let's see what we've got going on. Let's talk a little bit about what we learned last week. So last week you learned about scientific communication. There are all different ways that science is communicated, everything from news articles to journal articles to talks that are given at professional meetings to information that's provided by scientists through TED Talks and everything else. So there are lots and lots of ways that science is communicated. We also learned about the importance of determining who your audience is. And then when you look at your audience, what is their knowledge base? How much do they know about this? What are their preconceived notions? What are the questions that they're going to have at the end of this? What is their interest in this topic? So there are lots of things that you should consider when you figure out who your audience is, because you get to gear your presentation toward that group and then anticipate what they're going to ask you so that you can be prepared. So that's scientific communication. This week is all about biases in science. So scientists are human beings, duh. Anyway, we're human beings, and all human beings have biases. It helps us get through the world because we look at the world through lenses that we've developed through the way that we've interacted with our environment, and that includes the people in our environment. So we come up with these biases that we see the world through that particular lens. Well, scientists do this too. If you think about it, a scientist understands a whole lot about one particular area of science. And so that scientist has preconceived notions about what is going to happen if they set up an experiment. They have ideas about how this is supposed to work. So researchers come in with biases. There's something called confirmation bias. When you set up an experiment to give you confirmation that this thing is going to happen that you think is going to happen. But there are all kinds of other biases too, where you set up an experiment and you only use a limited number of subjects, or your subject pool, your patient pool is very small and it's very limited in age and race and gender, etc. And so you're going to get a biased result because of that. There's a famous study, and I can't talk about it in specifics or else YouTube will flag my video and I'll get kicked off of YouTube. But anyway, it concerned 11 young patients who had a disorder, and we usually talk about it on a spectrum, and they were also given shots in their arms for particular diseases. And the researcher determines that there's this connection between the two. Well, to start off with, he only examined 11 subjects, and they all were on the spectrum. Anything wrong already? Yeah, you can see that there's something wrong. But not only that, who was he paid for? So that's a very good question that we're going to get into. Who was he paid by to do this research? Oh, the lawyers of the parents of these subjects who are going to sue the people who made the shots. So, of course, he's going to find this connection, right? And so he lost his medical license. He can never even do medicine anymore because of this study that he did. And that's because of all the biases in it. Oh, and there were all kinds of other things. He doctored the data. Yeah, he made up stuff. Yeah, so there were all kinds of problems. So biases, we have to take them out of science if we possibly can. We have to think about this. Like, how am I going to make sure that I don't include any bias? And one way to do that is through double-blind studies. And that means that neither the researcher nor the patient, subject, client, whoever it is, knows whatever the treatment is in this case. The researcher and the subject do not know what treatment a patient is getting. Now, when I did the COVID vaccine trials way back in 2020, I was part of 3 different studies. This was a double-blind study, and so the doctor didn't know, and I didn't know what kinds of shots I was getting. And in the first trial that I was a participant in, I was actually given the placebo, so I had no protection. So that's a double-blind study. What bias does that take out? The researcher and the subject. So nobody knows what's going on until the end. All right, so that's one way to get rid of biases. But also, a scientist just generally has to think about all the different types of bias that may happen. Another type of bias that you may get is by who funds your work. I don't have that on this slide, but let's talk about that for a minute. Where is the funding coming from? If you work for a biotech company and they want you to do research on environmental effects of a particular GMO that they're selling, right, out to farmers, there's a lot of pressure on you to come up with no environmental effects, because who's paying your salary? The biotech company. Who's going to fund more research? The biotech company. So you can see how that would play into your bias a little bit. If you are subsidized by National Institutes of Health, which is part of the federal government, then, you know, they aren't expecting any particular result. So that wipes out that bias there. You always have to look at that, and that should be included at the end of a journal article. It always says that, what your financial interest in this particular topic in this study is. So that's really important. Look at the funding. Okay, then a couple of other things. P-value. So you want a p-value. P stands for probability. What is the probability that this phenomenon that you have discovered just happened by chance? So you want your p-value to be extremely small. You want it to be 0.05, which is 5%, or less. Sometimes people want a p-value of 1% or less. So what that's saying is that there's a 5% down to a 1% chance that this phenomenon that you saw was just purely happening by chance. What you're saying is that, no, this thing that happened is because of this thing that I tested in my experiment. So p-values are important. Confidence levels are sort of the opposite of that. So you want a confidence level of 95% or greater, and that means you have confidence that this truly happened the way that you said that it happened. And that brings us, because we're talking about statistics, and statistics are very important in science because it shows you what confidence level you have about what you found to happen. Gregor Mendel. So Gregor Mendel was an Austrian monk who studied pea plants, and he was interested in how traits were passed along into offspring. And so pea plants were very readily available because lots of farmers planted them in order to grow peas. And there were lots of different varieties of them. And so he grew, and they don't take up much space when you grow them, and they produce lots and lots of seeds. And so he chose pea plants as his experimental model. So what he discovered was that every organism contains 2 copies of every trait. Now, this was interesting because people didn't believe this back then. And there was no blending of traits. So it's not like you put the 2 traits together and they produce a new trait. That's not what he found either. He found that every organism has 2 copies of every trait, and some of those traits are recessive traits. And when they're recessive, then they can be hidden by a dominant trait. If you have a dominant trait, 1 or 2, they are always expressed. You always see the results of them. So he discovered this by studying pea plants. And this was way, way before DNA was even discovered. So how in the world did he understand genetics? He collected data on nearly 30,000 pea plants and their offspring. And he did these analyses to see if this is truly what was happening. And he figured out from the ratios that he got that every organism must have 2 copies of every trait. And that makes sense because you get half of your traits from your mother and half from your father, even if you're a pea plant. So now we understand this. Sorry about that. All right, then you're also going to read more about the Deepwater Horizon oil spill in 2010 and the importance of the science that had to happen. So for a little while there, we were depending on the BP scientists and the BP executives to make all the decisions about this oil spill. And then they were not successful in capping it. And so it was gushing day after day after day after day, millions of gallons of oil. And so the scientists had to step in. And when they examined the results, they said, no. There is 10 times as much oil coming out of this spill, out of this oil rig, than you are saying. So it's going to take a totally different strategy for us to shut this off. And so they did that, and eventually they were able to keep the leak from and keep the spill from just continuing on forever. But why was this important? Because the scientists had to step in. They didn't have anything in this game, but the BP scientists did, because the BP executives were saying, you got to minimize the damage, and it's the PR damage from this oil spill. So you got to keep down on the level of spill that's happening. So you can see that those scientists, even if they were scientists, they were under a lot of pressure to minimize the amount of oil that was coming out of the oil rig. So read up on that. That's very interesting. All right, then what is due this week? You don't have any discussion board postings. You have your rough draft. So you're going to take what you wrote in week 4, look at what I've graded after I graded, and make revisions to your week 5 rough draft. And you are going to include a hypothesis statement or section. It's going to be at least 3 sentences, remember, and then how a scientist would go about answering this particular question. So a hypothesis is usually worded as an if-then statement. So if this happens, then that happens, right? But I am perfectly fine with you just asking a question. Now, here's where you may be confused, because don't you remember in week 2, you came up with a question, and here we are asking you for another question. Okay, here's the difference. The question that you asked in week 2 was a big, overarching question. How carbon friendly are electric vehicles? How fast is the ocean becoming acidic? What's the effect of ocean acidification on marine life? Oh goodness, I can't think of another one. Are GMOs unhealthy for people to consume, right? You came up with a very big question that you could examine. You could go out there and look for research about it. The hypothesis is a much smaller and focused question where you could actually perform experiments to see what answers would be, right? So it has to be a much smaller question that you can actually design experiments to test. So you might want to say, what's the effect of changing the pH by 1/10 of a unit in ocean salt water on the calcium production of mussels? Okay, you see how it's a much smaller question, much more focused question, and that you could carry out in a lab. Now, you could carry it out in the ocean, but most likely you would do this in a lab. And you can figure out how the scientist would set up these experiments, right? That's the difference between your hypothesis and that question that you asked way back in week 2. Use your rubric to guide your work in order to do this particular assignment. This is a very important one because at the end of this, you will have gathered all of your information. Now, you won't have feedback on all of it. You won't have feedback yet on the hypothesis and the natural scientist section, but you'll have feedback on all the rest of it. So make your improvements as you go along, which leads me to the next slide. Yeah, use my feedback in order to improve your work. This is very important. And by the time we get to week 6, where you have to turn in your final paper on your topic, which counts for a whole heck of a lot of points, you need to have improved your work all along until you have basically a perfect paper, hopefully, in week 6. All right. With that being said, I hope that you have a great week. Let me know if you're having any issues, if you're having any trouble. If you were in the path of Hurricane Helene, which I was, I lost power for 9 hours there one day, but I've got all mine back on. If you're having any trouble and you can watch this video, please let me know. m.sigmon@snhu.edu, and we'll work around your situation. Okay? I hope that you have a wonderful week this week.